as we age, the livers cannot regenerate because they are getting older, essentially, right? The cells in the liver are getting older, and they can't replicate and divide as efficiently because of the telomere shortening and other reasons, just like we age, right? So we age and the cells in our body are getting old and reaching senescence. If you think about cell culture from last week, similarly, these cells, the liver cells in our body, they regenerate super well when we are young, but as we age, they lose that capability because they are aging as well, and they are losing that capability over time. And that capability can reduce later or earlier, depending upon the amount of toxins that they are exposed to. Because the more toxins they're exposed to, the more they're having to regenerate, the the shorter their lifespan's going to be, essentially, because they're having to reach that stage quicker. So because of that, we typically can do live donor transplant if the donor is usually like 50 or above, which isn't that old if you think about it, but it's old enough when you think about liver. So one of the things that people are looking at, researchers are doing nowadays, is trying to understand the cell biology, right? The cell cycle in a young liver versus an old liver and how they're different. And then they're trying to use supplements and other pharmacological means that people can take to prevent that loss of regenerative capability, or to kind of rejuvenate the liver, right? Make it young again, essentially. And in doing that, they hope that they can, one, protect liver longer, so let people have their liver stay healthy for longer, but also be able to use some of these older livers. If you have otherwise healthy donor and they are 50 or above, you can still use their livers for transplantation. Does that make sense?
Yes, makes sense.
So that's one part of the research. The other part is that there has been a big rise in acute liver failure, where patients, sometimes very young, show up to the ER in active liver failure without having any prior history of liver disease. And usually it's associated with overdose of painkillers like Tylenol, acetaminophen, or even they didn't overdose, but they've been taking it for a long time because maybe they were even sometimes very athletic folks. They're maybe hurt their ligament, or they pulled a muscle, and or something happens. They had some injury, so they've been taking Tylenol every other day or every day, and at some point, somehow their liver reaches this capacity and goes into failure. But the biggest reason would be usually overdose of Tylenol. That's one of the things we see. And in those cases, it's a very serious, life-threatening condition. And if you don't reverse it in time, it just causes death within 18 to 24 hours sometimes. Um, so they are trying to see if that can be either prevented, again, by taking some kind of supplement or some kind of a way to kind of save the liver. And so the same supplement is also being tested for this. In super emergency condition, that supplement has also been used in clinics as an actual ER as last ditch effort to save people. And sometimes it works, especially if the patient is otherwise young and healthy. They've been able to reverse it. So this case study revolves around this part of research and looking at that supplement and giving you some data to use for it, okay? So you can go ahead and read that in more detail when you do your actual case study. But it's looking at Tylenol overdose, and what they are using as model system are young mice, which are 2 to 4 months old, and they're comparing the effect of Tylenol overdose in those young mice to old mice, which are 24-month-old. So a 2 to 4-month-old mouse is essentially equivalent to like a 20 to a 30-year, 25 to a 35-year-old maximum human being. And the 24-month-old is like a 70 to 80-year-old individual. So that's the difference that they are looking at. And they're going to monitor it by looking at a protein, a single protein that is typically found in the liver. It is a very important protein in the liver and in its metabolic function called glutamate dehydrogenase. And the short form that we're going to use is GDH. And this protein is typically, you can't find it in the bloodstream, right? Or if you do, it's going to be at a super, super low level. But if there is a damage to liver, where liver cells are dying, actively dying because of liver failure, that protein is going to get secreted out as a byproduct into the bloodstream, and you'll see sharp increase in serum levels of GDH. So in the very first part, that's what they're showing you. They're telling you that they were looking at this effect, and they are looking at young versus old mice, and they're looking at serum GDH activity. Okay. So now, knowing all the information that I told you, you can think about when you would expect to see more GDH. Would you see it in young or old? And again, you can use the group chat or just speak.
So the GTH was the protein in the liver, right?
Yes.
You find it more in the young age, right?
No, so GDH in the liver is one thing, right? It's present in the liver to do work, that's fine. But you don't see it in the serum. What we are looking at is in the serum. And when the liver cells die, that's when you're going to see it getting secreted into the serum. So when would you expect to see more of it in the serum?
Okay, so in the old age, when the this protein goes in the bloodstream, pretty much, right?
Exactly. So you are going to expect to see it more in the older livers and in the older serum, because those are not able to repair themselves and regenerate their tissue as needed. So in this case, every single mouse that they are giving you data for, they have all been given a massive dose. So they've basically been given an overdose of acetaminophen, which is Tylenol, okay? And they are looking at 8 hours later, they're looking at serum of those mice. And they've taken exactly the same amount of serum. So for every single mouse, they've taken 20 microliter of serum to look at how much GDH activity is present in that volume. So it's equal volume, and they are able to look at it. Now, all the graphs in the case study are going to be where you can just simply copy and paste them into your Excel sheet. So what you would do is, so there are two things that I'm going to be talking about today. One is how you want to organize your Excel sheets and formulate your data, and obviously how to actually look through the data with the Excel and do all your calculations. But then also, how do you want to format and organize the Excel sheet as well as your figures? So those are two skills that we want to learn today. Now, when you are making an Excel sheet for your data set, you always want to keep it organized in a way that people not with any knowledge of the case study should still be able to open it up and understand what it is that you were looking at. So there are a few things that you can do, right? One of those things is that you want to label each sheet, and you can do that by right-clicking on the sheet where it says sheet 1, and then labeling it with whatever it is that you're going to be showing in there. This one is going to be young versus old, GDH, and I can even do serum GDH, right? So you write a little label for it so that anyone looking at it can know whether it was serum or tissue and what it was that it was looking at easily. And then I'm going to give it a label on top as well. Young versus old, serum GDH in response to overdose, or you can write the actual name of the salt, which is acetaminophen. I'm going to make it a little bit bigger so it looks better and more prominent, and there we go. Okay, now we have data for 1, 2, 3, 4, 5 mice per group, right? So our n number for this particular study is going to be n equals 5, right? And it's important to know for how many replicates you had. So you want to write it somewhere that you can keep an eye on that as well as needed for your analyses. Okay, so what can I do? What should I do first? If I want to analyze this data and I want to look at if there is a difference between young versus old, what can I do?
Can you repeat the question, please?
So I want to know if there was a difference in GDH levels between young versus old. What can I do to see that?
Bar graph, maybe.
I can do a bar graph. Should I do a bar graph with individual numbers or should I do some kind of computation
value.
first? What did you say?
I think with a mean value.
So with mean, average,
Yeah,
doesn't
average.
matter. Okay. So we can do that. So you already know how to calculate averages and standard deviations in Excel, but just as a review, you would say equal sign to tell it that you're putting in a function. And then when you start to write average, it should pop up. You select your numbers that you wanted to average, close parentheses. Do the same for the standard deviation. Select the same numbers and do that. Now, anytime we are doing our Excels, you want to make sure to not make them too busy. It makes it hard to see all the numbers and make any sense out of them, especially when you're looking at lots of numbers. Right now, just a few numbers, but sometimes you have data with 100, 200, 500 numbers. I've had a data set with 40,000 rows. I can't look at it if it had so many numbers, decimal points. So I reduce it down to one or two.
How do you do that? I'm sorry.
So I said you can reduce the decimal points by going up here to decrease it or increase it as needed.
Give me 1 second. I have a bunch of stuff open on my computer. It's so small. Okay, so you go to insert maybe, and then there is that. Or
insert what?
Where did you go? Oh, to home.
For what?
For decreasing the number of decimals.
It's up here on my little bar. But then if you don't see it up there in that top bar, you can go to format cells and you can do number. You can go to the number category, change the category to number and however many decimal points.
Okay, thank you.
Okay, so now let's make a graph. Okay, so I'm going to choose my labels, young and old, and I'm going to choose the averages, right? And if they are next to each other, that's fine. If not, you can do control. And while you're hitting control, choose the other two numbers, the averages, that row. If you are on an Apple, I believe it's command. And while holding down command, you choose the numbers that you want. And then you go to insert and we want a bar graph. So I'm going to go ahead and make my bar graph. Okay, so that's your basic bar graph. Now you want to format it according to appropriate way of presenting a graph. So this is what you would do. You want to add axis titles. You want to remove grid lines. Those are things we've just already talked about last week. And then you need to remove the border. So you will right click on the graph area, go to format chart area, and you would go to border and say no line. So you don't want a border. While you're at it, you will notice it does not actually have a line that is visible for your x-axis and the line for the y-axis, y-axis, and the x-axis line is very faint as well. So click on the x-axis and you would say, I want a solid line that is black. Same thing for your y-axis. I would say I want a solid line that is black. Also, this is super busy, this particular axis, so I can go into my numbers. So within your axis, you have these bars. You go to those You click on number, and I say, well, for this particular bar graph, I want it to show only 1 decimal place, and change it to that. You can even change the numbers so that it goes from 0 to 3, and the unit it shows are 1 apart, so it gets a little less busy. Then I'm going to remove the x-axis title because it already says young and old. I'm going to go to my y-axis title, and I will write what this is. And this is average GDH activity. You don't have the units for these, and that's fine. If you know the units, then you would change it to those. For the chart title, it is serum, oh, you want to write average serum here because this is in serum. So, and you can say young versus old, right? Serum GDH. Then you want to go ahead and change up your sizes of the text so that they are going to be better visible. So again, you're going to be home. You see it is at, the font size is 9. That's really small. Typically for our labels, we want the size to be 12 or 14 and bold when needed. This one as well, I'm going to make it 12 or 14. 14 might be too much for this one. I may make it 12. But yeah, it's fine, but it doesn't need to be bold. Same thing here, make it 12. Now, this one's good. I may make it bold. Now you have your young and old. You can leave these, but you can make it even more clear by clicking just on the young, and you can change it up. You can make the outline black. You can make the inside whatever color you want. Maybe I want it to be green to show they are young. And I can do that to the old as well. I can make them into a different color. Choose some mature color. That's better. Why not? I can do it that way. Then I can go in and I can tell that I want a legend, because obviously if I'm going to have different colors, then I need to tell people what those colors mean. And then you can move your little legend wherever you want. You can keep it up here in the top right corner. You can make it go straight across, whichever way you like. That's up to you. I typically have them on top of each other. There. And then obviously you need to change the font sizes in here as well to at least 12. There you go. And you can have it here, or you can put it up there, wherever you want. Okay, so that's your final graph up till now. And this is what we would call a properly formatted graph. It will have clear labels. It will have legend when appropriate. It won't have borders or grid lines, all that good stuff. The next thing we need to do is see if what we are showing is good enough, right? So what we are showing is average. However, which gives a good, some idea. It shows that old definitely has a little bit higher level, young is lower. But let's see if I were to plot all the individual numbers, right? The 5 numbers that I had, and show them side by side. When you look at this individually for the mice, you notice that in some cases, the numbers weren't that different, right? So if I look at n of the second round of my experiments, let's say these are, 5 different groups of experiments, the numbers are very similar. They don't show up as different. 1 and 4 show a lot of difference. 5 shows a difference a little bit, but these two don't. So we need to somehow, whenever we show our data, this would be correct, yes, but misleading. this doesn't show the whole picture. It doesn't show the variation between our data set. What can we do to show that variation? Because that is important. Otherwise, we are telling half truth.
Can we use the standard error bar?
Exactly. So we have to make sure that we are adding those standard deviations as error bars so that we can show that variation, right? So to do that, you will do the error bar, then go to more options. And we want both the positive and the negative. You go to custom, just like before, specify value, and then you are going to choose the standard deviations you calculated. Okay? And you would select those two and hit OK. And now they would be adjusted accordingly.
I have a question. When I did the standard deviation, I got different numbers. So I did the equal sign and then standard STD.
So I did the population number version versus you did the sample. That's why the difference.
Yeah. So population, that's what I have to choose.
For now, we are not going into when you use which one. You can use whichever you want, and they should both give similar idea. So you should be fine either way.
Okay, thank you.
Typically for sample, we need to have n of at least 30 to run that one.
That makes sense. When we only had like 5 now.
Exactly.
Thank you.
Okay, so far so good. Now we can see the variation, and we see that there is some variability within each data set, right? That the numbers weren't all like super close to each other. So we need to run some statistics. What statistics can we run? What would be a good statistics test to run when you have two groups to compare?
A student's t-test?
Yep, that's good. We can run student t-test. So when we are running student t-test, all we are doing is we are comparing two groups. It's a pairwise comparison, right? And that's what we're going to be looking at, the pairwise comparison. So what I'm going to do is show you how to do it the quick and dirty way, and then I'm going to show you a different way that you can run it in Excel. So the quick and dirty way is using the function in Excel for t-test. So I'm going to write t-test here, and I'm going to do it underneath this one. And for this one, I will say equal sign, and I can write t-test, and it will pop up. No, I did not write the whole thing. So t-test, and it's going to say what you want to compare. And I want to compare the youngs, which is my array 1, to the olds, which is going to be my array 2. And it's going to ask you one-tailed or two-tailed distribution. You're going to choose two-tailed distribution in this case. And then it's going to ask, well, what type is it? The correct way to do this would be to know whether you have equal variance or unequal variance. I'll talk about that first. If you do not know which one it is, you can run an f-test in Excel, which is also a function present there. Or if you are in a hurry, so-called, you can choose unequal variance as your default. That's going to be the most stringent of the criteria. So if you see a difference, then it is most likely present. The third option you have here is paired. Paired is used when the two groups are somehow related. That usually means that you have the same individual, maybe, a mouse that you took a serum from before you gave it Tylenol overdose and after you gave it Tylenol overdose. Or, the same cells that you're taking a sample from at various time points over time or whatever it is, under different conditions. Those would be paired. But if you don't have them paired like that, sometimes they pair humans when they have similar age and whatever characteristics they want to keep constant. But if you don't have related samples, they are not paired. In our case, they are very different populations, the young versus the old. Their cells don't even look the same actually at that point. So they're definitely not paired. And if you run the variance, you will find unequal variance many times. just because the variability in old is a lot more than young. So you would choose number 3 for that, and then hit enter, and you will get a number. Again, I'm going to clean it up a little. Now, does anyone know what number is considered significance or shows significance in a t-test?
Is it 0.05?
Yeah. So the p-value or the probability of this being incorrect should be 5% or less. So the p-value of 0.05, it has to be, p should be equal to or less than 0.05, indicates that there's 95% confidence that the data distribution you're seeing or the difference that you're seeing is correct, and 5% false discovery rate or 5% false positive rate. So that's what you want. That 0.05 indicates false discovery rate. And here it's 0.045. So is this significant? Based on this number, the t-test value here was 0.045. So does this show significance?
No. Right? Because
Less than
it's
0.05.
0.05, but this is smaller, right? So that means that
That
there
means
are
that yes, it's less than or equal to 0.05. You don't want a number higher than 0.05. Okay?
okay. Because I had this confused because I thought if the t-test gave us a number more than 0.5, that means that
Yeah, no, it's less. So you want 95% confidence in your results, right?
okay.
So that's not, that means 0.05 or less.
Okay.
Anything above would be less confidence.
Okay, makes sense.
Okay, the 0.05 is the chance that this is not correct. So it is correct.
Okay.
So if you find that it is significant, I usually highlight that cell to indicate that there is some significance, obviously. Another way that you can run t-test in Excel is by using their statistical package. You need to download that if you don't already have it. For non-Apple, so the non-cool computers like mine, you would go to Home or Files rather, I think Files, and you would go to Options. And under Options, you would go to Add-ins. Come on. And under Add-ins, you would do Analysis Tool Pack. And once you select it, you will right down here, it says Manage Excel Add-ins, and you will click Go. I already have them installed, so it shows a tick mark. Otherwise, you would tick mark, check in the squares you want. So we want the Analysis Tool Pack and hit OK. And it will show up under Data tab as Data Analysis. Okay, that's where it's going to show up. If you have a Apple, it usually is under something on this line. I think it's Options. No, it's not Options. It's Tools. It's under Tools on this top panel. You know how you have the File, Home, Insert? It's under Tools, and then you go to the same thing. You go to Options and Add-ins and everything the same way. Okay. So that's what you would do. Now, each part of your case study is going to have some questions for you to answer. The first thing you should do when you get the case study opened and when you're working on it yourself, you should read the case study and highlight all the portions that have the questions in it, okay? So when you read them, you will see that analyze the result and run appropriate. You can, why can't you use the same one? Whatever the answers are, just go ahead. The questions are, go ahead and highlight them so you don't miss them. Those are the ones you need to answer. The last part has two separate sets of questions. The first one is exactly the same as part 2. The second one is more elaborate. So make sure you mark both of them separately so you know what you need to answer. Okay? So just to show you what to do with these results once you got them, you will be turning in both the Excel sheet as well as a Word document with your answers and your data. And I'll be looking at both of them. I'll look at the document, obviously, for your answers and to grade you, but then I'll look at the Excel sheet to make sure that you're doing the calculations correctly and that you're organizing your document correctly. So you can open a blank document, right? And instead of answering here, you can answer, you can just insert an answer here too, but it's going to be a very long document then. And I usually copy paste the parts that I need to answer, or you can at least write the labels on each one, right? So you can say part 1 and write the questions, or just write the label of part 1 so you know what it is that you're answering. Now, to do this, it tells you for the very first one, it asks you, based on the data, did your hypothesis hold true? Why or why not? Explain your conclusion using graph and appropriate statistics, which is what you just did. So you need the graph, obviously, right? Because that's what you need to show. You're going to copy the final graph once you have everything on it. And you are going to paste it into your document as your figure. You're going to paste this as a picture to make sure all parts show up on there. And then you're going to add a title and caption underneath, okay? That here. Figure 1. The figure 1, the title is going to talk about what it is that this one is showing, the main conclusion from this particular graph, which is that old GDH serum activity rises in serum of old mice, or rises significantly in serum of old mice compared to young in response to liver failure, right? Now this is your title. This is giving you the main final conclusion, the take-home point of this graph. That's going to be in bold. In front of it, you will essentially be answering your questions as you write the caption, the details of this. So you will say young, 2 to 4 month mice, and old, 4 months. So you need to write basic information about what was the model system, what was tested, and what were the final results, right? So GDH, maybe, and you can write the, if you needed to, glutamate dehydrogenase, GDH activity, right, increased dramatically in, or in 4 mice compared to young to 4 month mice when they were given overdose, massive overdose of acetaminophen. The spellings are right, and then you need to write your p-value to indicate the significance, right? If you didn't run the statistics, you cannot write significant, the word significant in there. You can just say appears to, or appears to increase or have elevated, but you cannot say it is significant. To show significant, you have to have a p-value associated with it. And then you also need to write how many times the experiment was replicated. And you can write it here as n equals 5 per group, or you can be more detailed by writing a sentence or two about it. And that's going to be that. Now, any other information you need to explain that is not part of the caption, you can write your answer to that portion. Answer any other questions here, and then you're ready to go to the next part. Make sense? Now, when you're doing this, you're not writing like, paragraphs after paragraphs of information. I'm just looking for your critical thinking skills. I'm looking for you to look at this data and analyze it in a critical manner. That's it. So I just want a couple of sentences thought out and written about the data here. Okay? Okay. Next, you go to part 2. So again, in part 2, and I'm going to show you how to run the part 2, and then you are going to be doing the remaining stuff yourself, but you'll know all the skills. You'll have all the skills at that point to run it. Before that, let me show you the second way to run a t-test, which we hadn't shown yet. So you can go to the Data Analysis tab, and you can choose t-test. And in this way, if you're doing unequal variance, you would choose that one. And it will ask, what do you want? Well, variable 1, in this case, you want to make sure that you choose the label along with the numbers. So I choose the word young along with the numbers. Same thing for variable 2. And then I say labels are in the first row. And I tell it to give me the answer right here. I tell it where to give me the answer and hit OK. And lo and behold, it gives me my information. Now you can see it gives me the hypothesized mean. It gives you a lot of, it tells you, so the thing to make sure, it shows me the variance. It was different. It shows me the mean, which should be same as what you got for your averages. It shows me the observations, that there were N of 5 per group, and that's important. So you want to make sure of all of that. We are lucky that we have same number, but you can have different numbers of N's, it will still work. You can run a t-test. And then it gives me the t-test for one-tailed versus two-tailed, and both of them are showing significance. The numbers are going to be very, very close to what you got with your quick and dirty approach, which was right there. So this shows the 0.047 versus the 0.045 that we got. Still significant, still good. So that's the other way. Okay, so I'm going to do part 2. And then, you can have part 2 and part 3A, that little first part of part 3 on the same sheet. And then your third sheet is going to have your part 3B, which is going to be young versus old. Part 2 is just old, part 3 is just young. Okay, so now I write my label for this. It's going to be part 2, which is old GABA pre-treated mice. So the supplement that they are using is GABA, which is a neurotransmitter, but it's also found as a supplement. It's used for many, many different things. You probably know it as like a memory help or like mood booster, but it is also used in many anti-aging creams. It is also used by bodybuilders to build muscle. So it helps build and maintain muscle mass. So there are lots of uses for GABA. It's used in skincare products to like improve skin elasticity and condition. And so it has a lot of research as an anti-aging agent, right? Anti-aging product. And that's the supplement that we use in our research to look at rejuvenation of livers. So you can say GDH activity after liver failure in old GABA pretreated mice. Again, I'm going to make it a little bit bigger so it's nice and easy to see. Hold it, center it. There you go. Okay, so now I have the data in the case study itself. You will read about that. It will talk about GABA and how it is helpful. and it will talk about what it is that it's going to be using. This time, it's going to be looking at liver itself. And again, the same theory goes that livers from mice that are undergoing liver failure will have higher levels of GDH as they try to detoxify it and try to protect it when having trouble with it. And those numbers should go down when you give it GABA, if GABA has properties to protect the liver and help regenerate it. However, this time there's a problem. We are using tissue and not liquid serum. So we can't just take the numbers off their face value. So you've been given the GDH activity and you've been told that you had 100 milligram of liver tissue from each animal, and you changed it into lysate, where you basically burst open the cells and got all the proteins out. And then you took in aliquot, a small portion of that protein extract. So you took 20 microliters of the protein extract. Now, if we look at our stuff, right, where did the pen annotate? Yes. So let's say, I'm going to give you a hypothetical example to help you understand that I have two groups of individuals, two groups, and maybe, I'm taking samples from 10. So I'm giving you the average. And in each case, I have 100 milligram of tissue, okay, from normal patients, and I have 100 milligram of tissue from patients with liver cirrhosis. Now, liver cirrhosis is a condition where the liver is getting destroyed because of overexposure to toxins, right? It's getting necrotic, it's losing activity, it's not able to function anymore. And that's usually, if you look at the liver itself, you're going to have areas of dead cells, areas of inflammation, areas where there's a lot of necrosis and fibrosis and different things. And there'll be some normal liver too. But I took 100 milligram of tissue from the normal group from each patient, and 100 milligram of tissue from each patient in the cirrhosis group, okay? I dissolved that liver in 1 ml of buffer to make my protein lysate, to get my proteins, okay? So far, so good. And then I look at, let's say, my GDH activity or another activity of another protein of interest. And I find that my normal patients, I'm looking at some other enzyme maybe, and I see 500 nanograms or 500 units of activity in my normal liver, and I see 100 units of activity of my protein of interest, right, protein X. And I see 100 units of activity in my liver with cirrhosis, okay? So what conclusion can I make from this data? So this is normal, this is cirrhosis. Okay, what conclusions can be made? Do I have same?
Protein is reduced with the cirrhosis.
What did you say?
The number of protein units, I guess, is reduced with the cirrhosis.
They are reduced in cirrhosis. That's what you would get. Oh, yeah. And if you think that I needed replicates, this is average of 10 samples per group. And when I compare these numbers, I get a p-value of like 0.001. Okay, that's part of it too. That's it. P was less than 0.01, so super significant. Okay. Can I make the conclusion that cirrhosis had a lot less of this protein than normal liver? Yes, no, maybe.
Yes.
Anyone else? Nobody else is writing. Can some other people give a vote? Yes, no, maybe? So The answer actually is we can't tell. Why not? Why can't we tell? Why can't I tell? Oof, that's terrible. The hint to that is that I told you how the normal and cirrhosis liver were different. So think about the way I told they were different and tell me, why can't I tell if this number is correct?
Probably because the process of cirrhosis, it takes a certain time, and so like we don't have that information of like how long it will take for this difference, I guess.
Nope, we did get a particular given time for these patients, so obviously there seems to be something happening. Well, the reason is that, like I said, if you look at a normal tissue compared to, or you look at two individuals and you take the same amount of tissue, it's not going to have the same composition, right? So if you take people, a very athletic person's tissue versus tissue from a not so healthy person, it's going to have different composition of fat and different percentage of body fat versus muscle versus everything else.
Uh,
Same thing here. These have different number of hepatocytes at the end of the day. The 100 milligram of tissue from those two doesn't mean 100 milligram of hepatocytes. It means who knows how much of that 100 milligram was just inflammatory liquid in the cirrhosis or dead cells. We don't know that information.
and that's
So
the problem.
we don't know the total amount of protein that we extracted from these two. Okay? So let's say that I look at the total protein, I look at all the protein and I say I quantify it, and I find that I had 2,500 units of total protein, okay, in the first case, and I had 500 units in the second case. This is the total protein. Now, what conclusion can I make? Or not even units, let's write it in milligrams to make it easy for you. So let's say that I had 25 milligrams there versus 5 milligrams here. Which one had more now?
According to the numbers, the normal one looks to have more, but still, since it's tissue and we don't know the composition of it, probably the best answer is we can't tell.
No, now you can tell. When you normalize it to the total amount and you compare these 25 milligram compared to 5 milligram, the ratio would be 1 to 5, right? Cirrhosis has 5 times less protein than normal liver. Normal, right? And so when you compare those, they become equal. So to make it even more simple, it could be that I got 5 total milligrams out here and I got 1 milligram out here.
Okay, so you're saying by comparing those two results, we can make a decision?
Yep.
Oh, okay, that
So
makes
you need to have a total protein, so you know how much, So in everyday example, if I have 2 boxes with green and red balls, I need to know how many total balls I have before I can see the percentage of green versus percentage of red.
Yeah, makes sense.
Right? Last or a couple of weeks ago when we did the microscope images, same thing happened. You had to count the total number of cells, right, in the field to be able to get percent of cells that were dying in your control versus dead. versus Taxol. Otherwise, there was no way to compare because there were so many fewer cells in a Taxol setting. Right? Does that make sense, guys? Yes, no, maybe? So this is something that we have to do every single time. That any time that we are running protein or DNA or cell viability analysis, we have to normalize things to something so that, or equalize them somehow so we can compare between them. And we do them either by doing a relative comparison to control, which is what we'll learn today, or we can do what we did last time where we did percent of the total something, percent of total protein, percent of total cells, something like that, so that we can compare samples from two different groups that may not have the same N number at the end of the day. So here, because we are looking at liver, you have been provided with not only, where did the line thing go? The pointer, oh, who knows? You have been provided with the GDH activity, but then you've also been provided with information to calculate the amount of protein. And the way that's been provided to you is by giving you the absorbance of the protein, extract, and an equation to quantify the amount of protein that you extracted from there. Okay? So if you go back to the very, very, very first simulation, you had a standard curve that you drew so that you could quantify unknown proteins. And that's what you've been given. You don't have to make your own standard curve. You've been provided with the equation, but you will be using that equation to do the rest of the work. Okay? Okay, what do I do to go back to mouse? There you go. Come on, why aren't you moving? There you go. So in here, I guess I have to clear all my files. There. You've been provided with the equation and you've been provided with the data so you can calculate the concentration. Now, when we're doing standard curve, you have your independent and dependent variable. Which one is the independent variable in this case? Concentration or absorbance?
The independent is the absorbance.
Why?
Because we have a set concentration, right?
No.
Oh, okay, never mind.
In a standard curve, to make a standard curve, you would need to have a range of dilutions with known concentrations.
Okay, so we know the concentration, we don't know the absorbance, so that...
Well, in the standard curve, right? So I'm not talking about these numbers. I'm talking about the actual curve. You would need, right? So if you had a standard curve, you would need known concentrations, right? Now I need to write known concentrations protein concentration or whatever, right? In milligrams per mil. And you would need your y-axis would be your what is dependent variable, which is going to be the absorbance, right? That's going to be dependent upon the concentration because the more the concentration, the higher the absorbance. And that's how you get your linear curve, is by doing that comparison. And that's where you get your y equals mx plus b equation that you need to use. Okay? So knowing that, in this case, you have the y equals mx plus b equation and you have the absorbance. Where would you input this absorbance and which variable would you solve for, x or y? Hello, guys, nobody talking? So you've been provided with absorbance and you've been provided with an equation. Where would I input the absorbance, y or x? So look at the standard curve that I have drawn on the side. What do I have on the x-axis? Protein concentration or absorbance?
Protein concentration on the x-axis.
So which variable am I solving for, x or y?
Why?
No.
I didn't want to answer because I keep getting everything wrong.
I guess, and nobody else wants to answer. So what's on the x-axis?
It's a protein concentration.
What do I want to find about the liver lysates? Protein concentration or absorbance?
You want to find absorbance if you have the...
I have the absorbance right there. What do I want to know? How much protein was there? I don't know how much protein I extracted. That's what I want to find out.
If you have to put on the x, you have to...
I have to solve for the x, and I have to put the absorbance for y. So
Okay.
I need to use this number in place of y and then solve for x.
Okay.
Does that make sense for the rest of you that are mostly quiet? See, like I don't know if you're getting it unless you tell me, because I can't see you. In a real lab setting, I actually see your faces and I can say, nobody got that, did they?
Honestly, it is confusing, but I am following you still.
Yeah, so I just want you guys to remember every single time you think about independent variable and dependent variable in your experiment. The independent variable is always going to be on the x-axis, and the dependent variable, what the result is that you were looking for, is always going to be on the y-axis. So in a given equation, when you have a y equal mx plus b equation, you want to look at the data and say, have I been provided data for the independent variable or the dependent variable? And in this case, you were provided data for the dependent variable. And so you need to calculate the concentration of your proteins so you can use those. Okay?
Okay.
Okay, so now let's go further down and, I have to clear this again. Must be a way to not do that. And actually, if you go down below right below the data set, it gives you the instructions to do this, okay? So what you're gonna do is follow these instructions pretty much, but I will show you how to do that for part 2. You're gonna copy this, just like you did for the first one. You're going to take it here and paste it. I always leave a column empty in the beginning. It gives me space to write extra notes or anything else I may need to do, and it doesn't look super scrunched up, which I don't like. And then you're gonna go in under the data set and you'll find the instructions, and in there it gives you a template to follow. You're gonna copy this template and you're going to put that in your Excel sheet as well. And make sure these two line up, because that's going to make it a lot easier for you to put in your formulas. So that's the basic. Now, the first thing you want to do is change this sample name. I know it says the same thing, but you're going to change it so that it really says the same thing. And you'll see the importance of that in about 5 minutes. So equal sign, and I just click on the cell to say, I want that number, whatever's there, or whatever word is there, I want that word. And change that. Now what this allows me to do is that if I were to change this to names of the patients, for example, it will do it. If I left them some other way, it will, so anytime I make a change to my raw data, it will change appropriate columns in my analyzed data. And that becomes super important in real life research. And it protects you from accidentally cheating, for lack of a better word. It may be that you're tempted to change one of these numbers. Well, that's going to change everything downstream, and we'll know it. Okay, So next, what you need is that equation, because without it, you cannot calculate the concentration of protein. So remember that you were provided with an equation. See, it tells you quantify protein concentration in each sample using corresponding spectrophotometer readings and the provided equation. So we're going to take our equation because this is what we need. And this equation is the same for both part 2 and part 3. So once you have it once, you'll have it for both of them. There we go. You're going to rearrange the equation to solve for x. Right now it's solving for y, which is the dependent variable. But you've already been provided the y. You need the x. So you're going to rewrite the equation according to y, according to x, not y. So that's going to be the absorbance, which is y, minus 0.167 when you move it over there. And you can do this yourself, so I don't want to necessarily slow it down. And then divided by this m number, which is 0.286, and that's going to be your concentration. I am going to quickly, before I do anything else, clean this up so it is the way I want it. Center it, less numbers. There we go. Okay. The next thing you want to do is convert those numbers to microgram per microliter. The reason for that is because when you load your samples, when we do molecular biology, we have very small volumes. We don't have milligrams and milligrams of protein and milliliters of sample. We have very small numbers. Now to do that, this is actually a super easy conversion. How many micrograms are in a milligram? Anyone? How many micrograms are in a milligram?
A 1,000.
That's right, 1,000. And how many microliters are in a milliliter?
A million.
No. How many microliters are in a milliliter? Metric conversion, same thing. It's 1,000 again. So there are 1,000 microliters in a milliliter and 1,000 micrograms in a milligram, which means the answer to the next question is what? What should I write here?
Mm, 6,353
Divided by,
divided by 1,000.
which would be the same exact thing. So it's exactly the same exact thing. Because the ratio was the same, the answer is the same. So that's why it does not give the difference. Okay, the next question is, how much of this sample did I load into my actual activity assay? So we took 20 microliters of this sample, where each microliter has 6.353 micrograms of protein. So to calculate total amount of protein that I loaded into the activity assay, what do I need to do? I know how much is in each microliter. I want to know how much is in 20. What should I do? Come on, guys.
Divide by 20.
Why divide? I know how much is in 1. I want to know how much is in 20. So it should be multiplied by 20. This just means per, you know, PER per. So micrograms per 20 microliters, which is going to be basically, we know how much is in 1. We want to know how much is in 20. It should be 20 times as much. So you're just going to multiply by 20. Everyone with me so far? Yes? No?
Yes.
Everyone's lost. Do you guys hear all the sound in the background, or you're good? Am I the only one hearing it?
I don't hear any sounds.
Good. There's like a baby crying, my husband shouting at my other two kids, trying to get them to calm down. Nobody's listening. My nanny seems to be helpless. It's perfect. I'm wondering if other people can hear it. Oh well, they'll live. Okay, so next thing I do, this is where it all comes together, is that I need to find out how much protein I loaded in relation to each other. Now, the easiest way to do this is to make one of your samples arbitrary 1 or 100%, and everyone else is going to be either more than that or less. Does that make sense? So we are just going to use the very, very first control as our arbitrary 1. That's how much was our 100%. And so we divide every number that we will get in this entire study by that number. Okay? Now, to make sure that that number remains consistent for every single other row, what would I do? What can I do to this formula to make it a smart formula so I don't have to write it again and again? Nobody's answering. You guys remember the use of dollar signs? Yes, no? So we're going to add a dollar sign wherever we want to keep it consistent. Yes, lock the cell. We can lock the cell this way by adding a dollar sign to the J and a dollar sign to the cell. If I just wanted to keep the column consistent, but not the row, then I would just write the dollar sign next to J and not next to cell. But I want to keep the cell. I want to lock the cell. So I'm going to do dollar sign J, dollar sign 7. The next thing we'll do is do a GDH activity, which is exactly, it's the raw GDH activity. So it's exactly what's there. So equal sign, that number. And then finally, we're going to do a relative GDH activity, which is where we now take the raw number and equalize it by comparing it to the amount of protein we loaded. So we are going to take this and divide it by the relative protein amount. The first number should keep consistent. The rest of them are going to change. Once you have that row, you can drag and drop, and you have all your other numbers. So now, before I show you the statistics associated with it, I will show you my fun trick. First of all, so it's easy to see that this is from old. I'm going to copy this. No, don't copy that. Copy this. And I'm going to put it here. And I'm going to change it to 3, 8, and I'm going to write. Yuck. That's easy. Everyone good? Okay. Now I'm going to go ahead. I'm going to take it here. You want to go ahead and write it directly below the first one. And you'll see why in just a second. Okay, so directly below the first one. I'm going to then take this table and copy it. And I'm going to paste it right here. And this is where you see that why it was important for us to keep it consistent. It went ahead and changed all my calculations according to the new numbers without having to me redo all the calculations. So you can double check that by noting that this column, the sample numbers are the same, names are the same as this one. The GDH activity got switched over to this one. And so all the other numbers are going to be correctly put in now. The one thing that you want to double check is your relative protein, right? That it is switched to J27 and not the J7 that you had up here. So you want to make sure of that. That's the one thing that you do need to change. Because you see how the relative protein does not read 1. So this one, you just need to tell it what the new number was. So this was J27. And then everything else will remain fine. You just need to fix this as well. And it will fix those numbers. Okay? So that's that for your Excel for, how you are going to actually get your numbers. Now to do the statistics, I'm gonna show you for here. This time we can't run a simple t-test just by itself because we have 3 different groups. We have control, we have saline, and then we have GABA. So we need to change that information, right? To begin with, I'm going to go ahead and highlight the important information, which is the relative GDH. That's my final number that I care about. So I don't accidentally take the raw numbers, right? So you want to make sure they're highlighted. Yes, I'm going to post the recording. And then you will have, obviously, your, oh, why did it go? So I want to run them. Now, when you copy and paste these numbers, you have your 4 controls. You want to paste them as values and not as, just a simple copy paste, because you won't get the correct answer if you do that. Here, if I do that, it's going to give me that it can't do it because it copies and pastes the formula. So make sure that you do the numbers, okay? Just be aware of that. And then Control C. Okay, again, I always try to keep it clean a little bit for my own sanity. Here are the numbers. Now, to do the analysis, we can't run a simple t-test. We first need to compare all 3 groups together. So we need to run a different type of analysis called single factor Anova. Now, Anova by itself is not going to tell me if there was any, where the significance was. It's only going to tell me that somewhere in there, somewhere in these 3 groups or 5 groups or 10 groups, something was different. Some group did not behave like the others. And then you have to go back and run the t-test to find where that was, where it was that the things were different. So here, what I'm going to be doing is running the single factor Anova. Again, you need the data analysis tool pack for this. So make sure that you have the tool pack in there. And you're going to go to Anova single factor, and you're going to click OK. It's going to say what range, what do you want to analyze? The data has to be in columns with their labels. You select all of it, the labels and the numbers. And then you say labels are in the first row. And for the output range, you tell it that you want it wherever you want it. I try to keep it all together. So I'm going to say I want it right here, next to it. And then I'm going to click OK. and it's going to give me the results. Now for my results, let's look at those results and see what they mean. So in the results, what you should notice is that it has your groups correctly labeled. So it tells me the control saline GABA. It gives me the correct N number. So how many replicates I had, which was N equals 4 per group, and that's there. It gives me the averages and the variance, which is great. And then it gives me the p-value. And it does say that there is some significance somewhere in there. So that is what you want to highlight again, just like before. And now you need to figure out where the difference was. To do that, you need to run 3 t-tests. So control versus saline, control versus GABA, and then saline versus GABA. Those would be the 3 t-tests associated with this. For the actual data that you're going to be putting into the assignment, this is the table that you're going to be putting in along with the Anova results. That's it. The second and the 3A don't have anything else associated with it. You're just putting in this. So, you know, let's say it's or 2. And you're going to put that table in as a picture for part 2, and you're going to put the Anova results in underneath. So this is just your single factor Anova, and that should be enough for that, along with a sentence or 2 on what it shows. The 3 t-tests that you need to run are the other thing. And again, you can just run it the quick and easy way. So you can say t-test, right? And you can run these t-tests just like here. Control to saline, two-tailed, unequal variance, since we can see right here, the variance is different in most cases. And then I can change those according to what I need to show. So I can make this control GABA, and then I can show saline to GABA, and you have your numbers, and you know which ones are significantly different, so you highlight them. Okay, so these will be your 3 t-tests that you can label, make a little table of, and put that in here as well. You're going to do the same exact thing for your young data. The very last thing that you need to do, oh, I did that in the wrong one. This was and then this one is going to be part 3D. So for this very last part, now you are going to get a little bit more interesting. So in this one, you just had a single variable of the treatment, whether or not the mice got GABA, and then you just had the young mice or just had the old mice. In the very last part, you're now going to compare the results you got for part 2 and part 3. To do that, you have been provided with the actual template that you can organize your data in right here. Because this time, what you are going to get is a multiple independent variable data set. You have one independent variable, which is age, young versus old, and the second independent variable, which is presence or absence of GABA. Okay, so again, you label it accordingly. And to put in the numbers, you can just take the numbers right from here that you've been using, right? So you can do copy, and I can put those in here as values. And then I can go up to my old data, and I can put those numbers as values. And what this is going to show us is, this is going to look at two things. It's going to look at the effect of GABA in regenerating liver and rejuvenating it, making it behave more like the young. So that's one important part of the story. We see that it helps within the old group, but we also want to make sure it was making it similar to the young mice. So that's what it's going to be looking at. So these are your raw the numbers from what you had calculated before, and now you analyze them. In this case, I can't run a single factor anymore because there are two independent variables. So what you need to run is a two-way ANOVA. So once you have the data just like this, you go to Data Analysis tab, you go to two-factor, and you can run it two different ways. The two-factor ANOVA can only be run if you have the same number of replicates in all groups. We have n equals 4 in all groups, so we can do this. However, if you don't have that, the only way to run two-factor ANOVA is by taking the average of all your numbers and then doing it without replication. So not looking at all the variability, but just taking the averages and running it that way. However, we have the numbers, we have the replicates, they're all the same, so we can run ANOVA two-factor with replication. To choose, you're going to choose the entire area, everything in there, including the labels on the top and the side. For rows per sample, you write the number of replicates. So you have 4 young controls, 4 old controls, 4 young saline, so on and so forth. So you're going to write 4. And then for your output range, you are going to tell it where to put your ANOVA results. And you will hit OK. And these are going to be your two-factor ANOVA with replication results. That's the values that you got, right? Now, what do they mean? A few things to look at in there. The first thing is it's going to give you 3 different p-values, right? It's going to give you sample, column, and interaction. And you look at those numbers and you mark the ones that are showing significance, right? So I'm going to mark them, and then I'll talk about what they mean. When it's talking about sample, it's talking about comparing control versus saline and control versus GABA and saline versus GABA. So that's what it's talking about, the effect of GABA treatment within a group. When it's talking about columns, it's talking about difference between young versus old. So young control versus old control, young saline versus old saline. That's what it's talking about. And the last thing it's looking at is interaction. The interaction specifically is looking at this. And it's looking at whether GABA treatment affects differently young versus old mice. So if the numbers in here went down compared to control, but the numbers over here went up compared to control, then that would be considered significant. There'll be difference. But if the numbers in both these cases are going down compared to control, then or going up compared to control, so they're both behaving the same, you shouldn't see a difference. Now, we don't know if that's the case, because maybe young mice don't need regeneration help. Maybe they don't need rejuvenation. So maybe GABA doesn't help them at all or doesn't do anything more to them since they already regenerate. But this tells you that it does. And we can double check it by running the t-test between those two. This shows that there is no difference in how GABA behaves, how GABA acts on the cells, regardless of whether they are young or old. And that's an important distinction. So you already should have run these t-tests for your young and the old sample, right? The control versus S control versus G and S versus G. So those would be the 6 t-tests that you've already run. In addition, you would need to run young control versus old control, young control versus young saline versus old saline, young GABA versus old GABA, right? That's the important one, like I told you. Then you would also need to run young control versus old saline, young control versus old GABA. And that's important because this was the original question. Can GABA pretreatment help the old livers become young again? And that's what this one will show you, is whether it will become young again. So these would be the additional 6 t-tests that you would need to run to show significance, and then mark those and then answer them. And the last thing you would need to do is make a graph with your young and old mice, right? Young versus old, and control, saline, GABA, with their averages and their standard deviations. So that would be the last thing that you would need to run, would be doing your average and then the standard deviation and making a graph out of it. So you will have a single graph with 6 bars that are in pairs. So you'll have 3 pairs: control, saline, GABA, young, and old. And that would be the last thing you would put into the assignment. And write a little paragraph on what the final conclusion is and turn that in. So any questions on that? Yes, no, maybe? You can put it either way, whichever way is easier for you. You know, you can organize them this way or that way. I'm looking at the chat in just a sec. Sounds good. So please do make sure that you just email me if you run into any trouble, if you're having any issues with any part of the assignment. I will upload this lecture as well, so you have that information available to go through. But you have obviously the written instructions as well as the little tutorials from YouTube that are also inserted in the module that you can use for help as well. You guys are good with the graph? I don't think you should need any help. It's essentially going to be the same way with the standard deviations and everything, except it's going to look like this: young versus old. And then you can, with, you know, just the first 3. And it will say control, saline, GABA. And you can make all the youngs one color, make the olds all one color. Add the standard deviations and you'll be good. Okay? Sounds good. So that's all, folks, for today. Like I said, today was supposed to be the larger one. Davida, did you say something?
So thank you.
No problem. Take care. Bye-bye.